Thank you for joining us for the Soil Health Testing What We've Learned webinar. My name is Catherine Garcia, and I will be the moderator for the webinar. AgSource prides itself in delivery of quality, timely, and accurate analysis, providing information, valuable information and innovation solutions to improve decisions and profitability of members and customers. And we're so excited that you were able to join us today. Now, I would like to introduce our presenters for this webinar. Jim Friedericks. Jim Friedericks provides direction and support for the AgSource Laboratory educational programs, connecting AgSource with university and industry leaders. Jim has been with AgSource since 2012, supplying agronomic and environmental information to laboratory clients and professional audiences across the Midwest. And Stefan Gailans. Stefan is the research director at Practical Farmers of Iowa, a nonprofit organization that specializes in equipping farmers to build resilient farms and communities. Stefan has been with PFI since 2013 and leads their Cooperators program, which brings together a community of curious and creative farmers who take a scientific approach to improving their farms. Welcome to both of our presenters, and I will turn it over to Jim to get us started.
Thank you, Kate. It's good to see everyone, although I can't see you, but it's good to have you with us this morning. As Kate said, it's a cold and not very pleasant day here in Iowa, but we'll look forward to better weather, maybe even next week. I'm very excited to have Stefan joining us today. Just to give you some idea of the way this will flow, I'll give some presentation from the laboratory perspective on soil health and soil health testing and some of the research that we've done. Some of the research that we did has been in cooperation with Practical Farmers of Iowa. And in that regard, I wanted to make sure Stefan had an opportunity to present some of the insights that they've gained from their research as well. So I'll talk for a while, Stefan will have an opportunity, and then we'll have a chance at the end to answer your questions back and forth together. Just to give some context to what we're talking about today in terms of what we've learned from soil health testing. Soil health testing is one of the components of what we're used to in soil testing in general. Familiarity with soil pH and nutrient availability and CEC and organic matter, those are some of the bedrock soil testing parameters that we're all familiar with. And then there's also the quality aspects of soil, which would be the physical properties of soil, bulk density, and how porosity and water infiltration is affected by those physical characteristics within the soil and pore spaces that are there, the gas exchange that takes place. So those are traditional, we're well versed in all those characteristics and qualities of soil. Soil health is kind of new area. We've always talked about organic matter and how that affects the nutrient availability. And we've talked about organisms and their interaction with nutrients and how that benefits crops. But we're just now, in the last 10 years or so, really focusing more on the biological characteristics of the soil and how those microbes that inhabit the pore spaces and reside on the surfaces of the soil particles behave, how they interact with the organic matter that's there, how they utilize those nutrients and cycle those nutrients, and getting more detailed in how we can look at that information. But really it all gets back to organic matter and knowing how that organic matter interacts with the biology. Some is biological, actively biological, some of it's residual and resistant organic matter that is has more physical characteristics. So they're all related, and we'll all be looking at those interactions through this conversation, through this talk. Soil health relates to those living qualities of the soil. There's organisms that are there. There's billions of organisms in a gram of soil. And the diversity of that is it's overwhelming to even be able to catalog it. We're just starting to be able to do that and discovering more things as we start cataloging. And so understanding how those organisms behave, how they interact, is a developing body of science, but they do interact and they do behave in predictable ways. And so by understanding those predictable ways, we can manage those organisms. And it's very much like managing livestock on a farm. the soil microbial life is soil livestock. So they respond to inputs. Those inputs are food that comes from plant roots that are growing in the soil. As a plant captures carbon from the atmosphere, it transfers some of that carbon to the soil. Up to 25% of that carbon is moved into the soil and is used by those organisms in processes that we're familiar with, such as nitrogen fixation or the symbiotic relationships with mycorrhiza and the benefit that is for both organisms. And there's also parasitic relationships and mutually beneficial but not symbiotic relationships as well. So the interaction with roots and plants and living organisms in the soil is important, but there's also the residue from biological life organisms, biota that die in the soil, as well as plant life and plant material that's deposited either on the surface or under the soil surface from root mass. So it's all these are inputs into the livestock that's there, along with air and water and temperature and physical parameters that the soil provides. That's the living structure, that's the milieu within which these organisms survive. And that's what we're trying to understand when we start looking at soil health characteristics. AgSource Laboratories started doing soil health testing in 2016. This is the report that AgSource has developed. Other labs have been in the business about that long, some longer. And the testing that they've evolved has followed different tracks. We adopted the Haney soil health protocols. And so this is what we generally offer in our laboratory. Of course, there's other approaches to that as well. On any typical soil health report, you should see the soil testing characteristics, fertility parameters, pH, organic matter, CEC, nutrients, pK, calcium, magnesium, etc. On this report, you see that on the right side, on the left side in the blue. And then there's a recommendation for corn. In this instance, what fertilizer would be required to produce corn on that soil. And then in the brown area, the green and brown box on the right is a soil health analysis. Today, we're going to look primarily at that top box, soil health score, Solvita CO2 respiration, which we no longer call Solvita, we just focus on CO2 respiration, and the CDN ratio and how those interact. Though the respiration and the CDN ratios are the primary components of the soil health score. And so I'll be explaining that a little bit more. We'll explore how those interactions relate and talk about that both from my perspective as a lab as well as Stefan's perspective in field applications. When we started soil health testing in AgSource, we wanted to get a handle on the variability that we're seeing early on and how that progressed through the year. So we did a very simple study each month. One of our staff went to a field out near his home, and on one side of the fence was a pasture, and the other side of the fence was a field that was previously in corn, and the year that he sampled, it was in soybeans. And so every month he sampled this. The green dots are the cornfield, and the blue dots are the pasture. There's a lot of up and down in those dots for each month, and what was most confusing was the fact that the pasture was more variable than the row crop field. So this just left more questions than it had answers. And so we decided in the next year, in 2018, we would do a more comprehensive study. And this is where I started a relationship with Practical Farmers of Iowa. They had a number of research projects, and I was able to piggyback along with one of their projects in Southwest Iowa on a grass-fed beef farm, Dave Schmidt's farm, and we were out there able to collect samples from his farm 4 times during the growing season. The area was split into 4 different areas. The PFI was primarily interested in the grazed and the ungrazed CRP land, but I was interested in the differences between the row crop and pasture land. And so we did all 4 of those. Each of those locations had 3 specific sites that we sampled every time we went out and sampled. So at each location for that gray CRP, you can see 3 blue dots, the lighter blue ones. And I went back 4 times a year and collected 10 cores at each of those locations. So each one of those was one specific sample. So in a year, we collected 4 times 3 locations, and just collated that over the 3 years. So this is a study that was designed to be able to statistically say what's the difference between these management strategies, what's the difference between time of the season when they were sampled, and the growth stage of the crop that was growing there, and then what's the difference between positional location of each of those 3 locations within each sampled area. One more thing to note, the main pasture was right by the farmstead. It historically was manured, so there's a lot of manure history on that location. The grazed CRP is most similar to that in the type of soil, same soil series up on top of a ridge. The row crop and the ungrazed CRP were most similar soils as well. The row crop was the flat bottom and had been in row crops for a number of years. And the ungrazed CRP was just up the slope a little bit above that on the crest of a slight ridge. And historically, I think it had been cropped, but now it was in CRP and had been for at least 5 years before we went out and sampled. These are the images, pictures of those locations, the row crop on the left and the pasture on the far right, and the CRP in the center. The soil tests of those were significantly different. You can see that the row crop tested lower in almost every category particularly than the pasture, and also than the CRP ground. Low pH, low organic matter, very low phosphorus and potassium, and the lower CEC in the row crop area. The pasture, as I said, was manured historically. Organic matter, the unit, more than a unit percent higher than the row crop area, much higher native phosphorus and potassium from all that manure that had been applied over time, and a higher CEC. And again, if you look at the row crop and the ungrazed CRP, those 2 soils are more similar, and the pasture and the grazed CRP, those 2 soils are more similar in characteristics, particularly the CEC and organic matter content. Bulk density also indicative where there have been a lot of manure and pasture for a long period of time, lower bulk density than the row crop, which had a higher bulk density. On the right side is the soil health score from those areas, aggregated over the 3 samples and 3 years, and for all 4 season sampling events. So you can see statistically, row crop has the lowest soil health score, and the pasture has the highest, and all of them are statistically different as we went through these things. Looking at them aggregated or grouped differently, the average soil health score by crop stage, which was early June, end of May or early June for V4, if it was a corn crop, and then July, the second stage is R1 for corn, so just at silking. RM would be black layer for corn, and then samples time 4 would be post-harvest. And 2 out of 3 times it had already frosted before we sampled, so that reflected a dramatic change in the season. So although there's some variability in that graph, they're not statistically different, just because there's quite a bit of variability between those, across all those different management practices. And then from year to year, they're a little bit different, the 3rd year being a little bit lower than the first 2 years in soil health scores. Again, that's probably weather related. 2020 was a drier year than the prior 2 years, and that seemed to affect the values that we got. The components of soil health score it's a calculation, and it's based on the CO2 respiration, which is, I'll go into that in a little bit more detail later, but that's a flush of carbon dioxide as we look at the sample when it gets re-wetted, and then the carbon to nitrogen ratio based on water soluble carbon and water soluble nitrogen. So there's the equation from that. Usually the values are between 5 and 50. Those are typical. If it gets over 50, by the way we calculate it, that indicates there's an excess of soluble nitrogen. And in this calculation, we include the nitrate or ammonium as part of that soluble nitrogen, which helps to identify those really high values and gives you a higher score. In this case, over 50 doesn't mean it's a good score. It indicates there's a reason for that higher number. The carbon and nitrogen are analyzed together in a water extract. Shake some sample up with water, filter it, and then read a total carbon and nitrogen concentration with a carbon CDAN analyzer. Just to get a context for these CDAN ratios, a typical well-decomposed organic matter, the organic matter portion in the soil would have a CDAN ratio of about 12 parts carbon to 1 part nitrogen. Soil organisms tend to be a little bit lower than that, on average around 10. Bacteria are low, 5 to 1 or 8 to 1, whereas fungi tend to have a higher carbon to nitrogen ratio of 7 to 1 all the way up to 25 to 1. The higher CDAN ratio indicates that carbon is able to grow and thrive on, as you know, on wood sources, and utilize the little bit of nitrogen that's there in their metabolism as they grow. And then for comparison, wheat straw would have a CDAN ratio of about 80 to 1, something like that. So that we talk about that in terms of nitrogen mobilization and mineralization and immobilization when we think about adding nutrients, adding carbon sources to the soil. Looking at those factors from these different locations in that experiment, the row crop soluble carbon is lower than the pasture. In fact, it's one of the lower carbon values. And on the right side, the soluble nitrogen follows the same pattern, not surprising. The CDAN ratios would anticipate that. And if you look at those numbers, the carbon numbers are roughly 10 times higher than the nitrogen numbers, which bears out in that typical CDAN ratio, which you see here. On the left side, the CDAN ratio is around 10, except for that ungrazed CRP, which had a little bit more carbon in relation to the nitrogen that is available in that site. And these are aggregated over the 4-year or the 3-year study. So it's a real difference. But CDAN ratio is not always a really good predictor of what's going on in that field in terms of soil health, because if you look on the right, you can see that by the variability by the time we sampled. So the first May and September are statistically different than July and November. And the ratios vary because of the variability of the carbon and of the nitrogen that happens in that soil. If you look at that more specifically, on the left, that CDAN ratio by crop and by month, you can see what's going on in the row crops. A low CDAN ratio early in the spring, then it goes up as the crop grows. And incidentally, the 2 years that we sampled the row crop area, the first year was corn, second year was soybeans, the third year was corn. And so, and this is the average of those 3 years. Then at maturity, that CDAN ratio drops again, and then post-harvest it goes back up. That pattern's repeated basically on all the different management locations. But if you look at the carbon and the nitrogen separately, you can see there's different kinds of trends that happen. Carbon tends to increase for each of the locations as the season progressed. In the row crop, it went down, but then it went up at harvest. Whereas when there's a consistent cover crop on the field, that soluble carbon tended to increase throughout the growing season and maximized after the harvest period or first frost. Whereas the nitrogen was higher in the spring and then went down when the crop was actively growing, and then started to come back up again at maturity and then dropped again later on after harvest. Not unusual. You expect, especially in the row crop, to have a lot more nitrogen available early in the spring after fertilization, and then it's utilized, and then it gets re-released as the crop matures and decomposition sets in after harvest. So those variabilities, I think, looking at the seed-end ratio, it gives you an indication of what's going on in terms of the metabolism, but really to get an idea of what is that metabolism doing, what's happening with the organisms. It's instructive to look at both the carbon and the nitrogen amounts and see how that relationship relates to what's going on in the both with respect to the crop or plant that's growing and the organisms that are there in the soil. The respiration rate is measured with what we call a burst method. We take a dry sample, rehydrate it, and then seal it up in a vessel, incubate that for 24 hours, and then read the amount of carbon dioxide that's respired by that moist soil after 24 hours with a infrared carbon analyzer. So we get an actual measure of the carbon dioxide carbon that's produced by the organisms in respect to that metabolism, that rate of growth, that rate of utilization of the carbon and nitrogen that's in the soil in that 24-hour period. The respiration rates, again, you can see the variability. Statistically, the row crop have the lowest respiration rate. The others are not statistically different. The value somewhere between 60 and 150 is what we typically see. The row crop is trending on the low side. The others are showing good respiration rates, as we would expect from having a consistent cover on the field. And that, that's one of the things we hear often, is that the longer you can have growing vegetation on the ground, the better it is for soil health, and this kind of gives an indication of that. I'm going to turn over to Stefan. He's going to talk about some of the projects that they had with on-farm trials. And so thanks, Stefan, for joining us, and go ahead.
Thanks, Jim, and thanks everyone for being here. Like Kate mentioned at the top, I work for Practical Farmers of Iowa, which is a farmer member organization. Our members have been conducting on-farm research since the late 80s. And I'm going to share a couple of examples from last few years, that are similar to the project that Jim just shared, that he was directly involved with. So you can go to the next slide, Jim. In this first example, we had a group of 4 farmers who wished to test their soil's health among 3 different fields on each of their farms. So each field was in corn soy production, but the fields differed in that one had no cover crop, one had a cover crop that was not grazed, and then one had a cover crop that was grazed by cattle. So each of these farms did have both crops and livestock. Each had 3 fields on the farm, and the cover crop in question each fall was seeded to cereal rye. They wanted to know how cover crops or grazed cover crops affected their soil's health. And so to do that, we helped them by designing an experiment. Soil samples were collected in the spring of 2019, 2020, and 2021, as well as in the fall of 2019 and 2020. So just like the experiment Jim just showed, we sampled these fields multiple times over the course of the years. They were sampled to a depth of 6 inches at the same point in the field each spring and fall. And then we sent the cores to AgSource for analysis to determine. We looked at soil respiration and water-soluble carbon, 2 of the metrics that Jim has already talked about. Okay, so what did we find? Next slide, Jim. Okay, so this is for soil microbial respiration. You can see average across all sample dates, we compared the no cover crop field to the crop field, or the no cover crop field to the grazed cover crop field at each farm. So the graph on the left are the results from each farm when we compared no cover crop to cover crop. In other words, how did the presence of the cover crop affect soil respiration? So in this graph, to the left of that 0 up and down line indicates reduced respiration on account of the cover crop. To the right of the 0 line would indicate improved respiration on account of the cover crop. And so then we see a red notch for each farm. and error bars around or about each notch. And the error bars help us determine whether the change that we observe is statistically different from zero. So if an error bar overlaps with the zero line, we consider the change to be statistically zero. In other words, no change. Okay, and then on the right are comparisons between the no cover crop field and the grazed cover crop field on each farm. And in this figure, in this graph, we see that one farm, the Friedericks farm, that's the second from the top, did see a statistical improvement in soil respiration. The error bars for that notch do not overlap zero. But interestingly, we see at the Degner and Kennedy farms that soil respiration was statistically reduced in the grazed cover crop field. But taking a look at the means in either comparison shows that we can't detect a statistically negative or positive change in soil microbial respiration compared to no cover crop, at least with the data that we collected over this time period and at these farms. Okay, so next slide, Jim. As I mentioned, we also looked at water-soluble carbon. And just like with the previous slide, we came to a similar conclusion with water-soluble carbon as soil respiration. Neither the cover crop field, that's the comparison on the left, or the grazed cover crop field, comparison on the right, had a significant change compared to no cover crop on average compared to the cover crop. So interestingly, again, the Friedericks farm on the right shows a vast improvement in active carbon, water-soluble C compared to no cover crop where they grazed cover crop, but every other farm saw no statistical improvement or change. And the mean was also over all the farms, over all the time, no different than zero. So that's what we have with the data that we collected. Not entirely sure why, but I do have some take homes at the end that we can reflect on. Okay, so next slide. And finally, I wanted to share one more project that involved a similar setup that came on a little after that other one began. This was on one farm in Northeast Iowa. Again, it involved 3 fields on that farm, no cover crop without grazing, and then a grazed cover crop. And this took place over 2 years. We sampled at the farm April of 2020, and then again, April of 2021. So you can go to the next slide, Jim. And on the left are the results for soil respiration, on the right, active carbon or water-soluble carbon. We do see that in April 2020, on the left there, microbial respiration, the grazed cover crop, that's the green bar, did score best among the 3 fields in terms of microbial respiration. But the next year, we come back a year later, April 2021, and we were not able to detect a statistical difference among the fields. The little dots within the bars represent each individual sample, and you can also see in April 2021 at each of the fields, those dots kind of are a little further spread apart than they were in 2020. So that just reflects the amount of variability that came from each of the samples that probably reduced our ability to detect a statistical difference among the 3 fields. Similar story with active carbon, water-soluble carbon on the right. In both April, similar story to the previous study, excuse me. In both Aprils, we were not able to detect a difference among any of the fields, at least for that metric. So taken together, the results of these 2 studies have led me and some others to conclude that, the time of year, the year itself, and the location can really affect soil metrics. And I think that was reflected in some of the stuff that Jim shared earlier. And that can really affect soil metrics, and maybe more so or just as much as the practices in question. So I'll reserve some final comments and take-homes at the end, like I mentioned, and I'll let Jim get back to the rest of the presentation. Hopefully we can catch up at the end. So thanks.
Thanks, Stefan. I just have one more thing I want to talk a little bit about on this study, which again, the 4 different sampling times in the 4 different locations and management practices. And I kind of agree with Stefan on the, excuse me, the difference really the most dramatic differences that we saw was in management practices. And that seemed to be, there's a lot of variability in time of when the samples were collected, but the real differences that we could pick out were in management practices. Excuse me. So we'll look at, just to remind you of what the values are from those things, and you can see that row crop is significantly different than everything else, basically, except for maybe the CRP grazed land. The bottom line there is mineralizable nitrogen. We're able to make a prediction of how much nitrogen could become available from mineralization of the organic matter it's in the soil. It's a stoichiometric equation, but it does a good job predicting what kind of value, what kind of nutrient availability could be there historically. Excuse me. We've talked about organic matter providing roughly 10 pounds of nitrogen per 1% organic matter. And so that's an estimate that we've had for a long time. Here is another number that kind of validates that, especially on the pasture and the CRP ground. Those values are fairly similar to what you see in the organic matter component. Excuse me. The equation for mineralization is based on respiration rates and a function of the C-to-N ratio. It's actually a quadratic equation that affects that, that is coupled with that respiration rate. So what happens is if the C-to-N ratio is too high, meaning there's a lot more carbon than there's nitrogen, then that value can go to 0 because the negative factor of that first part of that equation. Even if it's a high respiration rate, it can still end up with very low mineralization of nitrogen because of the high carbon content. Or the other side of that is if the respiration rate is too low, it can also go to 0, indicating that there's just not enough microbial activity, even though the C-to-N ratio may be closer to an ideal value. There's not much biological life there to utilize that and take advantage of that. Based on that that equation, this is again that graphic of the mineralization rates of those fields. The low organic matter soil, the low respiration, the low soil health, everything about that row crop location indicates that it's not going to provide much nitrogen from mineralizable sources, whereas the two CRP areas that are similar, the pasture and the grazed CRP, are able to contribute quite a bit of nitrogen from mineralization of organic material. Interesting though, the ungrazed CRP was similar to the row crop, probably because that C-to-N ratio tended to be higher than the other 3 locations, and that just moved that equation farther to the left and lower C-to-N ratio or lower mineralization rates. As we look at soil, one of the reasons we picked the location that we did on that farm was we could find locations that were close in proximity and similar in soil type, although they ended up being two distinct soil types. But there are other things that affect soil, primarily the availability of nutrients and the availability of moisture and aeration. Those are mitigated by or controlled by texture, structure, organic matter, and aeration. One of the best places to look at those differences is, as you may have guessed, on a golf course. We have run, AgSource has run soil health samples with golf courses for a number of years. And this past spring, last fall, we compiled all that information and tried to differentiate or discern what's going on with that data. And particularly, we looked at the 2 different extremes, the fairways, which are more typical of the landscape, and reflect the soils that are there, and they're under high management, but they're reflective of the natural landscape. And the greens, which are primarily engineered soil, high in sand content, low organic matter, and they're under very high management, high nutrients concentrations, as well as water regimes that are managed, very carefully. So we took all that data and looked at the median values, which would be the 50% in the center, high being the upper 25%, low being the lower 25%, and looked at the distribution of these scores within that median value. So organic matter at the bottom, 2 to 3.5% organic matter in landscape fairways. That's what you'd expect. Soil health scores came back 20 to 40, which is right smack in the middle of the anticipated or the optimal range, which I put there in parentheses next to the categories. So the respiration rate is pretty high. The soluble carbon is pretty high. The soluble nitrogen is pretty high, or average to high. So these are just indicators of what we have been talking about generally with typical soils. But when you look at the manufactured soil, the Gulf greens, very different picture. The median values are about half of what they were in the fairways, tending to be on the low side. So if we use the same scores, same ranges on the Gulf greens that we use in the fairways, the situation, the recommendations, the interpretations would be, well, these Gulf greens are in poor soil health. That may not be the case. We looked at all the greens that we had, and these median values, this is the typical sample, and it constituted a lot of 1,500 samples, something like that, in the combination of both of these. So it's a large set of data, and I think we can make some good interpretations from that. But the take home from this is soil makes a huge difference, and the management of that soil makes a huge difference. So whether you're handling typical soils and pushing it towards higher productivity, such as in horticultural applications, where you're applying a lot of nutrients, a lot of amendments, or you're in a rangeland where you're not adding anything and trying to manage that, each of these will have different targets, and maybe you don't want to compare one against the other. Maybe you just want to compare them internally to themselves. Interesting from this is also the mineralizable nitrogen. Even there's still a fair amount of mineralizable nitrogen in that Gulf green, just because there's a lot of respiration, a lot of activity going on, and release of that nitrogen into the soil. To see that graphically, just quickly, you can see that the fairways virtually are twice the values as the Gulf greens, just to get a graphic of that. So managing soil health is a function of those things we can't control: climate, landscape, the soil properties, things that are inherent in the location, but then the things we can control, like plant cover and the amount of percent of time that there is a plant growing in that soil. That influences the diversity of the microbial population, and helps to support that population by giving inputs into that soil. There's carbon that flows from the atmosphere into the soil, as well as the nutrients that we apply to maintain plant health also influence the soil health as a component, as part of a whole cycle that we're managing. So think of it potentially as managing a carbon cycle, not just the specific nutrients, but the whole cycle of carbon utilization by the plant and by the soil, as it's influenced by these soil characteristics, soil and climate characteristics. So thinking of carbon then, and looking at how carbon, there's a lot of talk about carbon sequestration and increasing the amount of carbon in the soil. And I think one of the obvious, maybe not obvious, but one of the insights that we really need to keep in mind is that carbon respiration is tied to carbon cycling and carbon accumulation as well. these are two graphs of the two sites that I indicated. The one on the left is that 3-year study. You can see that as the organic matter increased from left to right, the respiration rate in that soil also increased. It's not a real strong statistical relationship, but there is a trend there that you can see. That same trend is true in the Gulf Greens, even though the organic matter content is much lower. It's only up to 1.6%. There's still an increase in the CO2 respiration as that organic matter in the soil increases. Whether you measure that as organic matter or you measure that as total organic carbon from a combustion analysis, that carbon accumulation cannot take place unless there's also a increase in the respiration and that turnover of carbon in the soil. That's just a component of the carbon cycle. So we need to keep that in mind when we think about growing our carbon stocks in the soil. We also need to be growing or improving our soil health, because the two are intimately related. Another example of that is a series of samples that come from potato fields in Wisconsin. These are highly managed fields. I mean, a lot of crop rotation, but the cash crop is potato crop. So everything's focused around maintaining high fertility and low disease pressure on that potato field, and maximizing the production of that potato field. So it's very intensely managed. You can see that these are measured by total organic carbon rather than soil organic matter. But that same relationship, I express the values as percent organic matter from calculating off that organic carbon. And that same relationship is there even under these highly managed field situations. When you have high organic matter, you get higher respiration. And if you want to maintain that higher organic matter, you have to make sure that there's enough nutrients and carbon, particularly, to manage that respiration rate for those organisms. Of course, that comes with a higher nitrogen availability and that cycling as well. So those are kind of the take homes from my perspective. Is soil health is a component of what we've traditionally known about soil. We've talked about soil testing and the qualities there. We're all familiar with how that works, but the soil is more than just a reservoir for nutrients. It's also a reservoir, a resource, and a physical environment where organisms, including plants, grow, and they utilize that space to take advantage of the aeration and the water, and the storage of that in the soil, as well as the components of that soil, the nutrient components. But the soil health is kind of ties it all together, and it's this respiration, this utilization of nutrients, the recycling of nutrients, the availability of both carbon and nitrogen. All those things play a strong impact on that soil health and the cycle, the availability, the release of nutrients back into the soil from the organisms that are there. So ultimately, it's organic carbon that we're talking about. And if you keep that in mind when we think about carbon sequestration, carbon credits, you can't do that. You can't focus on that alone. It also has to be built on good soil health and soil management that will promote soil health. Stefan, back to you.
Great, thanks, Jim. Yeah, I just wanted to circle back to some of the things that both I've kind of found, and I think Jim mentioned as well in some of the on-farm studies that we partnered on, and that some of the things that I consider the take-homes from those experiences, and that we saw quite a bit of variability across farms and the production systems that those farms were engaged in. And so probably not surprising, even though these farms might be all in Western Iowa or in a county apart, they still are subject to different kind of climate and soil factors that might affect these kinds of new soil health metrics. Certainly variability across years and within a year. Jim showed some of that from the trial involving Dave and Meg Schmidt's farm that involved the CRP. That was a really interesting thing to see how things can fluctuate through the year and how that might be different in a perennial field or a row crop field. So, that's something to keep in mind about, oh, is that good? Is that bad? I don't think we know whether that's good or bad. It just is. And I think going forward, anyone wishing to, test their soil health, evaluate their soil health, using a local reference as a benchmark is important. There's been a few studies in the scientific literature that have kind of reflected that as well. It really gets you a sense of what's the potential of your soil at your location, so that you're comparing against yourself or your region rather than against a farm or a farmer or a management practice or a system that's not applicable to you or your location, or the enterprises that you choose to raise on your farm. And then finally, I do think it's Jim and I kind of talked about 3 or 4 year studies here. Probably need to track these things a little longer. Some of those odd patterns that I mentioned or Jim mentioned too, those might smoothen out or reveal themselves to be more of a pattern if we can look at things over time, and rather than just kind of comparing it, A versus B once during the year or once over time, and really, then using that to measure your field. And are you seeing progress in your field, not compared to someone else, but compared to yourself? I think those are the ways that some of these tests, at least the way I see it from what we've been doing, are most applicable, at least at this point in time. So I'll leave it at that.
Thanks. I think, strong take homes. There's no bullets. It's not a silver bullet to get the best yields or anything like that, but it's a long process. So we're at the end of our presentation. Thanks for listening. We do want to open it up for questions, and you can direct your question either to me or to Stefan. I think Kate will host that part of it. Thanks.
Yes, thank you both to Jim and Stefan. One housekeeping thing before we get started on the many questions that came in. You can see on the screen is the QR code for the CEU credits. Go ahead and scan that to get immediate access to the CE credits. We will also be sharing that QR code in the email that we're going to be sending out to participants. So we are going to go through the questions in the order they came in, and I have notes on who was presenting at that time so we can direct them to that person. The first question we had came in was for Jim in regards to how do these soil health tests compare traditional soil fertility testing and providing recommendations.
The soil health tests, the Haney soil health test, which is kind of what we use, incorporates the traditional soil fertility components, which we offer that, but then the respiration and the soluble carbon and nitrogen, those are uniquely biological. And then beyond that, we do offer the availability of nutrients based on a weak organic acids, which is the H3A test, if you're familiar with Haney's terminology. And those give an indication of some of the nutrient availability that's available to the plant root at the time of sampling or at the time of testing. Whereas traditional soil fertility test gives you that, but it also may be more of an indicator of what could be available over a longer period of time. So recommendations that come from that, the fertilizer recommendations are based on traditional soil fertility testing that we do. We, at this point, recommend some management practices. You may consider adding more legumes or fewer legumes to your cover crop to get a better seed-in ratio, better nitrogen availability in your soil. And then further recommendations would be consider no-tilling versus conventional reducing tillage in some way and trying to add more cover crop to your rotation. The longer you can keep crops in the field, better it is for soil health. So not a lot of specific recommendations come out of soil health testing, but more general recommendations. These are some practices you can follow that will improve over a long time, will improve soil health.
Thank you, Jim. The next question is for you again. Did you use a soil probe or a spade?
The samples we collected were collected with a soil probe, 6-inch depth, and we collected 10 cores at each sample site every time we went out and sampled. So one location had 10 cores, and then we moved 30 feet or 30 yards away, connected the second sample and then the third sample, and went back to those same locations every time we sampled. But they were the cores, yes.
Great. And then again, how does the ratio change when you have a cover crop grazed in the spring and fall and have a row crop during the summer growing season?
I'm assuming that'd be the seed-in ratio.
Yes.
Yeah, it depends. Seed-in ratio is dependent on the fluxes of carbon solubility, your soluble carbon and soluble nitrogen. And so in the row crop situation, we saw that carbon tended to decrease through the growing season, probably because the just the ground cover was minimal and the available carbon was being utilized by organisms that are there until it started to be depleted. Whereas under an area with a cover crop or that's grazed the cover crop, that carbon would be continually supplied by the growing plant. And so there's more nutrients, more carbon availability over time. So it's hard to pinpoint what would happen to the ratios because both nitrogen and carbon fluctuate. But I don't know, maybe if you have an insight on that, Stefan, you're welcome to join in.
Oh, I don't, but not looked at it. So I think you made a good stab at it though, Jim. I don't have anything to add. Yeah.
Great, thank you. We also had a question come in cover crop systems with good amounts of residue, should we anticipate a release of nitrogen late in the growing season to offset some commercial nitrogen needing to be applied side dress? Maybe you'll, yeah.
I would say yes. I would anticipate as that organic layer, as that organic material that's residue that's decomposing, it should release. Of course, it depends on the kind of residue, but if it's high in nitrogen, meaning it has a low carbon nitrogen ratio, it will release nitrogen that will become available as it decomposes. If it's a very high carbon component like straw, wheat straw, something like that, it'll decompose more slowly. It may tie up nitrogen initially, but over time it will release some of that nitrogen back in as it decomposes. So it depends on the material that you're incorporating or leaving on the surface.
Thank you, Jim. The next question is actually for Stefan. What were the Degner and Kennedy farms perhaps overgrazed and that led to poorer respiration?
Yeah, well, I can't say for sure, but I know the farmers involved in these studies are really vigilant about when their cattle should be on or off their crop fields grazing to avoid pugging or compaction. So I don't think it would be, I wouldn't go so far as saying that the overgrazing maybe affected the results, but certainly could be if a field were overgrazed. So I can't say for certain one way or the other than that the farmers we work with are pretty vigilant about when it's appropriate to have cattle out on crop fields grazing in the fall or spring based on conditions. So yeah, that's what I'll say to that.
Okay, thank you. The next question came in when Jim was discussing the final slide about the joint study for soil health. You had mentioned management practices. So such as what kind of management practices?
Well, it was, Sorry if that was confusing. I was referencing the different locations in that farm. One was the pasture, one was the cropland, and then the grazed and the ungrazed CRP. I didn't point it out in the slide, but one of the slide where I had those little blue dots, the grazed CRP also had a series of darker blue dots, which were added in the second year and in the third year. So we did sample areas that have been, so the first year was grazed in 2018, the second area was grazed in 2019, the third year area was grazed in 2020. So we had one location that was grazed 3 years prior, and the other two were sampled in the year they were grazed. We didn't see, and I didn't look at it really carefully. Maybe Stefan remembers how that turned out in their look at it. Didn't see a strong influence of grazing on the values of this, any of the values in that grazed CRP location.
Great,
Yeah,
thank you.
Jim, you're correct that the influence of the grazing in the CRP was not really detectable after the grazing compared to the ungrazed CRP. Yeah.
I think it'd have to be a longer term study to measure that than what we did.
Okay, the next one we have like a cluster of questions from a participant. So how would you evaluate application of a biological versus an untreated check, and how would you use the Haney to compare?
I could take a stab.
Yes, take a stab. I'm confused by the question a little bit.
Well, I think so. That's a kind of thing that farmers I work with ponder, and typically we then would suggest they run a replicated strip trial, alternating strips of treatment, no treatment, or in this case, biological and non-biological. And really, to me, and what farmers I've worked with have shown me over the years is the true test is, well, what's the yield and ROI at the end? To see did that product or whatever that intervention did for the overall productivity of those strips, and did it pay for itself, or was it an added cost? I think you could take that to the next level of saying, okay, I'm going to use this product, and can I reduce any fertilizer under my typical rate, to see if this product or whatever intervention has an effect. You could, I guess, you could do soil sampling. Another take home that I kind of find on these large commercial fields is that, as Jim and I kind of both saw, sometimes we don't detect differences where we might anticipate, or we've been told we're going to see differences. And sometimes I think that it reflects that we can't take as many samples as maybe we need to detect things with some of these metrics. So ultimately, I think, a strip trial and see how it affects the crop yield at the end of the year and the returns on investment are a really good, easy way to see, is this thing worth it to do anything for me?
Thank you, Stefan. And the same user has a question: is water soluble C in Haney and label active C in POXC a permagate and total organic carbon? And can you explain the difference?
They're related. They're not the same analysis. The POXC is a permanganate oxidizable. It measures some of the physical stable components of carbon and actually oxidizes them with the compound, whereas a soluble carbon measures what's immediately available, immediately active, because it is soluble in water. It's a very quick extraction. You shake sample with water and then filter it, and so it's not a measure of anything that's potentially or going to be available with a little bit of chemical activity that takes place, which would be a typical biological reaction. So they're very related, but they're not the same thing.
And then, have you compared the Haney with the new soil genomic tests?
I think the genomic tests are great. Their advantage is they can identify those organisms that are doing specific things. And if you can demonstrate that there's a sizable population that's utilizing or utilizing some compound in some way, nitrification or denitrification, whatever it is, if that population is dynamic and large compared to other populations, that it's a predictor. It's not a measure of actually what's going on, but it's a good predictor of what is taking place. These soluble availabilities, the respiration rate, those are predictors as well. So they're different ways of getting at some of the same information, and each has its very strong advantages. I think coupling genomic testing with that is another good way of estimating what's going on in the soil. So I think it's a great improvement, a great advance in our understanding.
Thank you, Jim. The next question is, my clientele are very interested as humic acid as a manure and fertilizer replacement to increase soil health and soil nutrient availability. What is the viability of adding humic acid for soil health and or fertilizer replacement?
Humic acid it definitely does affect the soil. It does improve nutrient retention and some of the availability of nutrients. The advantage of manure, the advantage of green manuring is that it captures carbon, it captures other things besides just the physical, what the humic acid brings, which humic acid is just one component of organic material, the humates. But whereas manure and plant residue brings in a whole host of other components, other things as well. So yes, if you can find ways to get humic acid to provide what you're looking for, I think it's great. I think it can be in addition to or supplemented by other practices that are also encouraging nutrient retention. One of the advantages of cover cropping is nutrient retention, not just organic matter accumulation, but also capturing those nutrients before they're leached from the soil, going into the subsoil and bringing up those nutrients and retaining them. And so there's other aspects of sustainability and fertility management that we think about when we think about manure and nutrients and cover cropping in that respect. So they're all tools. They're all things that we can use to improve crop production, and I think each has their benefits and needs to be understood in that way.
Thank you, Jim. And I'm going to answer one last question. We still have a few left. So any questions that we did not get to today, we will be responding to you guys personally via email to answer any of those. So the question we're going to end the day with is, based on the variability of soil health testing throughout the growing year, when is the best time to test for soil health?
The same time every year. That's a cheeky answer, but because of the variability, it's hard to say one is better than the other. I would tend towards either spring or fall. Spring is helpful because it gives you that predictive value, especially if you want to use that nitrogen mineralization value in your fertilizer rates and applications. So spring would be a good time to do that. Fall can do that as well. It doesn't account for some of the changes that happen over winter, but it's also a good time. But as I said, it's good if you do sample early growing season in 1 year and you want to monitor what's going on from year to year to year, just be consistent and try to pick conditions that are similar to what it was when you sampled the first time. So if it happened to be, really warm and really dry that first time you sampled, and the next time you sample it's cool and wet, those values are going to be different, even if it's a similar time of year. So keep in mind a lot of the variabilities that happen, because it's a biological function, it's a biological process, and it does change based on what's going on out in the field. But springtime would be probably my preference. Spring and fall would be the two options.
Great. Thank you so much to both of our presenters, Jim and Stefan. We really appreciate all of the insightful knowledge you gave us today.